Good afternoon. So this is Pankaj, and I'm going to take a brief on Genes2me. First, a disclaimer that we are Genes2me India-based company, and we are into real-time PCR, molecular biology, and NGS-based panels to create for. And today's my talk is to give you a gist what we are doing right now and where we see how we can support the scientists and the community for the better worlds. So presently, what we are having is we are having solutions for oncology, human genetics, and infectious diseases. Infectious diseases we are developing more. From the morning, you would have been seeing what are the important roles of NGS and how they are transforming. But I like to highlight what the challenges also one's face when one go through with all these newer technologies. Number one, always keep changing the hardwares and the technicians are difficult to get and train for. And then you also have a capital investment and maintenance whenever there is a new upgrades are coming. And so also the compatibility of the skilled staff to compete with the same kind of upgrades what is happening with that. So keeping in mind these side effects, what is more important to my brain is the TAT presently and its implications, and not only for the sake of experiment but also to do the analysis and give the data for the patients at the end of the day. So presently one can see this is the technology. It has changed the paradigm and everybody wants to go for whole genome sequencing. But the challenge is how much depth and what is the analysis capabilities and how we can transform that to clinics as soon as possible. This is a major challenge which we have seen when we were trying to see how to implement this in the clinical world. And then what we have concluded is targeted panels, especially onto the oncology and the areas where huge genes, large number of genes are involved. That areas with the larger size of the genes could be more important in the targeted. And the major thing which we noted where we are seeing all the people are representing from the cfDNA for the targeted panels, what is the coverage? And the major thing to our brain is whenever we speak about cfDNAs, wherever we speak about somatic mutations, the coverage plays a very crucial role. As more and more you do, more better conclusions you can make by doing higher coverages and also the intelligent selection of the genes, which presently everybody is doing. Now, how to select the genes? How to select the panel was the first question. We thought, okay, we concluded targeted panel is the way to go. But how do we select panels? How to go about and where to see for the literatures? The seminal paper from the cell way back in 2011, where they have concluded what are the different pathways that are being involved for making the a cell being cancerous and what are the different genes involved with that. So with those clues, we started putting onto our efforts that thinking onto the same lines of the pathways, can we dig more literature and collect what is being happening in the current world and how we can support the current world with the known therapies and the FDA's, how the process goes on to make the things happen at the clinical world. On the second hand side, when we speak about hereditary disorders or rare genetic disorders, obviously rare always goes for the whole exome level. But when we know something about it, can we go a little more deeper and intelligent selection so that the analysis could be fast tracked, automated, and without much interventions, the thing could be translated immediately to at least clinically significant mutation reports. That was the aim of doing this. So this particular diagrams are available from the cell, and these are being taken also from epigenetic metabolism paper, which you can always refer back. Now, what we have concluded in the recent time is now we are able to translate those basic information way back published in 2011 to the level where people are coming up with more and more kind of drugs being targeted towards different pathways, towards different metabolisms, how we can stop, and then how do we apply that to happen. Every other quarter, we are seeing more and more number of drugs being targeted and available to see for the researchers, but still it is a challenge until unless it comes to an FDA and somebody has some proofs to do a trial, or at least to the people who are not responding, we can do that. I would like to start with a classical paper which got me introduced to this personal medicine, paper from Nikhil Wagle way back in 2011. This is the person who is having the melanoma, and you can see all over the body. And when they have taken the biopsy and they have understood that this is B600E mutation, and they at that time they have used PLX 4032, and they have seen a very good results that everything disappeared in 15 weeks of this particular treatment. Unfortunately, 7 to 8 weeks later, he redevelops it, it relapses. And at this time, he was kind enough to again give back the researchers the biopsy. And what we understood was the other mutations, what was happening and how to go about. Unfortunately, this patient passed away after 2 weeks, as per the paper suggests, but at least it threw the light how we can do the analysis based on the present failures also, and understand how we can improve for the next generation. So what we have understood from this is personalized medicine is promising, as we are seeing in figure B, and then resistance will develop obviously, and can we have elucidated mechanism to understand how it will develop, and can we do the beforehand itself the combination therapies or the ways to handle the management, so that at least it can be a better, longer, better life for a patient than going with a less one. Although many people have suggested, I have taken the example of malignancies here just to give you an example. What we have done as studies is looking at different levels of MDS and see how these myelomas are different and what genes are common and what is available as an FDA kind of reported drugs, and other drugs which people are trying to see in the generations where people are trying to look for effectiveness of them. What we have done is we have selected the ones which are more common, and also included the ideas of having some specificity so that one can do a common level analysis, as well as the classification based on the genes what you have on the list part. And with that logic, what we have come up is we have come up with 49 genes for this myeloid leukemia. And if you are seeing the people, my earlier presenters who have presented, you have seen all important genes. Very important gene in these lines are CEBPA, which is supposed to be GC rich, but very good marker to see the classification of. FLT3 gene, IDH1 genes, JAK and other genes are so important. And this all in a single way, we can do the analysis and to a depth of the requirement as per the requirement of 500x to 1000x minimum that is possible at very normal stage of doing the job. So with this, the idea is to keep the size in such a way that the first problem of ever-changing hardware can be handled. And this can be used on any machine, right from MySeq to all the way up to bigger ones. Of course, with the smaller chip itself is good enough. On the second hand, it also has an applicability of other like Thermo and MGI based on the adapter sets, what we are changing. The same gene sets can be performed on any of the machines, what is available in the market right now for second generation part. So in the similar manner, we have also supported not only for looking at the genes, but also the software. The major bottleneck presently is how do we analyze and report the software? And we have put more efforts in developing CLISI interpreter software. This is cloud-based software, and it takes both routes of somatic as well as germline analysis. And this takes approximately 5 to 6 hours per sample. And we are trying to manage a cloud server in such a way that whatever be the pressure on us for number of samples, we should be able to work between 48 hours to have a first round of annotated BCF. And then once the phenotype information is more trained to the software, it gives you clinically significant mutation report. And based on the question you are asking clinically, whether you want to do a classification, whether you want to see which drugs are more targetable or which, how do we handle the kind of combined therapy, that can be seen with this kind of software as well, based on the phenotype and the question you are trying to ask for. With this logics, what I have done here is I have just listed all different panels what we have created in the oncology space, right from the single gene BRCA. So we have generated pan cancer, which is our favorite panel, 524 genes covering all important MSI, TMB, as well as looking into two of the important viruses, so that you can have a risk as a proto-oncogene that can be covered as well. In the inherited disease panel, we have also focused on to clinical exome sequencing, where we are having 30, close to around 7,513 genes, with 19.6 MB. Again, this can be applied onto any second generation instrumentation that can be done for easily up to 100x coverage at very kind of effective manner to look for. For personalized medicine, we have got pharmacogenomics panel. Metformin standard panel covers 122 genes with a small size of 524, which you can use for looking into which genetic makeup will suit a person for which kind of drug he is taking. On the liquid biopsy side, presently with the NCCN guidelines, we have covered ctDNA, colorectal, breast, and lung. And here the focus was to idea behind is relapsing and response to therapy. We understand that there are few things in the market for screening, close to around 300 genes which people are trying to screen with the fusions. We are working on those lines, but the number of depth requirement and the sensitivity, we are trying to work it out. At least with the blind studies, what we have done, we say we need to do little more. That is why we are taking little more time for the screening level. But we feel that relapse and response to therapy with these small set of genes can be easily studied and can be monitored effectively on those lines. For the disease specific panel, for those who are interested in the genetic, kind of disorders, from the cardiovascular, neuromuscular, as well as looking into some of the skin disorders and bleeding, we have got a lot of other panels that are being covered to look for. Especially, lastly, I'll mention metabolic disorder panel, where this is a single gene disorder panels, where we have combined 71 different genes to give you an answer what kind of single gene disorders that can be screened in a single go. So the idea behind for us is to provide you with a technology which is scientifically up to date. So we are working hard. My team is working hard to have whatever new genes come in, and that's how we will be able to add on to the future next version levels if that happens. Ours is hybridization based technology. So that makes the life simpler. By one tube itself, you will be able to do number of targets from few hundreds to up to few thousands. We are also using enhancer technology because the only limitation which hybridization technology has was the time it takes to do the hybridization process, whereas the amplification goes much faster. So we have generated something like around 4 to 6 hours. We can do the job efficiently for small number of size of the panels. That takes care of this little bit faster TAT. Within 1.5 days, you can still generate the data. We also provide pre-capture pooling. Presently, we recommend up to 4 samples that can be pooled together, so that you start with the individual library, you put the index and barcoding, and then you do the pooling. So while doing the target, enrichment, you will be doing 4 samples together, bringing the cost down. So also maintaining the sensitivity and effectivity of the panel in the similar fashion. We are trying to work the algorithms in such a way that GC panels, 80 GC rich regions, 80 rich regions are being captured equally. And the similar example which people have shown, CE-BPA, we have seen that at least when we are looking into the 500x coverage of those, we are able to get to at least 200x coverage of even the challenging genes which we have listed, 4 or 5 genes to have our internal studies being done. Analysis software from FastQ. So you will be providing to the software, the feed data comes in the form of demultiplexed FastQ file. And from there, you will be able to generate an annotated VCF. If you want to do your own phenotype analysis, you're welcome. But if you want to throw that to software, which is able to do clinically significant mutations, that can also be performed and given as a report. And lastly, just to again reiterate that these panels are compatible with mostly all instruments from Illumina, Thermo, and MGI. And just to share the upcoming stuff from us, we also collaborated with Element Biosciences, which is coming up in a big way for looking into the things. Our panels are also getting, kind of started to get validated on as well to work with. With that, I would like to say that we are able to give you the better data, ability to perform with the better things, and the genes, which probably you also gets connected once you see the genes list that we have listed what you are trying to look at. With that, I would like to thank you all, and I would like to ask any questions if you have. Thank you so much.